Hello. In this tutorial, we would like to show you an easy and fast and accurate way to analyze time-lapse movies such as these. In this movie, you see adherent cells moving in a chemotactic gradient. The gradient was performed with the microsite chemotaxis by ibidi. And the movie was put together by taking images every 10 minutes for up to 12 hours. And now we would like to analyze in which direction those cells migrated in this chemotactic gradient. So what we need to do to analyze this assay is single cell tracking. And now we would like to show you a fast way to do single cell tracking, which is using the platform FastTrack AI. FastTrack AI is an online cloud where you upload your image series or time-lapse movies and can download the results after an artificial intelligence-based algorithms interpreted those or analyzed them. And this is the home screen of FastTrack AI after registering. You can register for free, and you also get free vouchers for uploads, so-called analysis jobs. And after you redeemed the vouchers, you have a certain number of analysis for free, or maybe even purchase the voucher, then you have a certain number of analysis of jobs that you can upload. And for everyone new to FastTrack AI, we usually recommend to start with a quick analysis wizard, which guides you through the individual steps on how to upload data and how to download them again. The first step in this quick analysis wizard is giving your experiment a title. Let's call it chemotaxis test in our case. So then you can also give it a description. And as you can see, FastTrack AI can offer tools to analyze 3 different assays, cell-based assays like tube formation, wound healing, or chemotaxis. We go with the chemotaxis because we would like to go do single cell tracking, and then the next step is to click Next. This is the second step where it asks you what would you like to upload. Single images is usually an endpoint analysis, but as I've shown you, I would like to analyze a time-lapse movie or an image series that was used to generate a time-lapse movie. So in my case, I used the microsite chemotaxis from ibidi and would like to upload all the 3 wells I performed on one slide in one go. So I have 3 wells, and each well contains the images we've taken every 10 minutes. So this is time point 0, 1, 2, 3, and so on. And you can see how the cells are moving from image to image. So this I would like to upload. I have 3 wells of those. So what I choose, since it's a time-lapse experiment, I select time-lapse. Then you have to determine the time between the images. In my case, it was 10 minutes. And then it's important to indicate the microscopic resolution for your microscope and your camera. How to calculate it is given here under the question mark. So we find an instruction how to calculate the microscopic resolution. And this we need to get reliable data on the cell migration speed, for example. Then next step, it asks you how you organized your data. In my case, I have my information per well. Each well contains the images in one folder. So I have my files are organized by folders, one folder by well. So I click that and go next. And here we are on the uploading page. So all we need to do now is to mark the folders I would like to upload and drag and drop them. Then you click Analyze. And then the data gets uploaded, which takes a minute or 2, depending on the size of your images, the size of your movies that you would like to upload, because you can also upload time-lapse movies, and the number of wells or movies you would like to upload. You will get an email notification as soon as your upload is finished and as soon as the analysis is available. And as soon as it is available, you find the data under projects. So now we go to projects. And here you find for first-time users the quick experiments. The quick experiments is the folder where all data that you uploaded via the quick analysis wizard is stored. So therefore, our experiment is in quick experiments. This is our test. So we go there. And since we only uploaded one replicate of this test, my 3 wells, all done on one day, I have to have a look at those with a magnifying glass. And here you see all 3 wells are uploaded and analyzed. And to get a report with all the data, with all the results, you go under reports onto the magnifying glass. And now the report is loading. And this is the experiment report. First, you find some information, but the most important part is in the last tab. It shows you the results of the individual wells. So this is well 1. And I showed you that I uploaded images, image series. And the artificial intelligence-based algorithm now took those images, recognized every single cell, and followed each and every single cell from image to image to image. And by this, it put it together afterwards into a movie. And here you can see the tracking lines of the cells that have been recognized and tracked. It performed this for all 3 wells. And of course, you get lots of further information and data like a trajectory plot, where we see in which direction each and every single cell moved towards. You get information on the cell velocity, and another presentation of the tracking direction. You even have a glossary here on this page with all the information you need on how to interpret chemotaxis data and how to analyze single-cell trackings for chemotaxis assays. So it's quite convenient to have the glossary on the same page. And of course, the FastTrack AI is also comparing the results. So we uploaded 3 different wells, and the comparison you find under View Wells Comparison Results. And here you find the data and comparisons of all the 3 wells. And for example, for the forward migration index, you see definitely a difference in one well, which is a good thing because well number 1 and 2 were my controls, and the actual experiment was well number 3. And again, center of mass displacement is also quite obvious that one compound that I tested was chemotactic. And of course, you can also download the information. So you simply go here, click, and a CSV Excel file will be downloaded, or many Excel files actually. You also get a text file with the tracking lines or for the tracked cells per well. But I think the most important part is the summary that shows you the comparison of all 3 wells with all the information the system gathered, like FMI, forward migration index, center of mass displacement, directness of the cell, of the migrating cells, and even some statistics, the Rayleigh test. So FastTrack AI gives you results in a very fast and easy way. You simply upload your data and download the results. And with this, I wish you fun testing this platform. Bye-bye.